So in our last video recording, we followed electrons from plastoquinone. As it becomes plastoquinol, it picks up two hydrogen ions, moves through the thylakoid membrane to the luminal side, where it becomes oxidized as it reduces iron-sulfur complex. As it becomes oxidized, it releases those two hydrogen ions into the lumen of the thylakoid. The electrons then, of course, move on through the electron transport chain, one at a time through plastocyanin until each electron is used to reduce the oxidized form of P700, and those electrons have been moving through photosystem 1. Two have to collect in order to be picked up by ferredoxin, and ferredoxin transfers those two electrons to NADP+. So we talked about all of those steps there. So it's a good idea to go back through and review the steps just to make sure that you understand how many electrons are moving at a time, when two electrons are, when an electron carrier is waiting for two electrons, and also to pay attention to the collection of hydrogen ions. So we have two hydrogen ions that were moved from the stroma into the lumen here. We have two hydrogen ions that were released from each molecule of water. So really that ends up being 4. Once we look at two water molecules, releases 4 protons, which we were emphasizing before, plus the 4 electrons. And we're going to see here how we can accumulate more hydrogen ions in the lumen, and we'll come back to that here shortly. But ultimately, these ions accumulating in the lumen are then going to be transported through this ATP synthase complex here. So the electron transport results in the synthesis of NADPH. The accumulation of hydrogen ions inside the lumen is what fuels ATP synthase to produce ATP. So we're looking at this reaction here, ADP plus inorganic phosphorus or phosphate, to produce ATP. And this is an endergonic reaction, and so this reaction requires the input of energy, which is fueled by the proton motive force created by the hydrogen ion gradient, such that the concentration of hydrogen ions in the lumen is greater than the hydrogen ion concentration in the stroma. So that concentration gradient of hydrogen ions is what then builds up potential energy, and then that potential energy then drives this complex to bond the phosphate groups to ADP and produce ATP. So the movement of electrons through the electron transport chain is to NADPH is about 32% efficient. So it's 32% efficient of energy transfer by electrons to NADPH. And the remaining 68% is used to basically synthesize ATP by moving or shuttling hydrogen ions across the membrane in various ways, is then ultimately used to make ATP. So the synthesis of ATP is one of those critical outputs from the light reactions. ATP is used in the Calvin cycle, which we saw earlier in our sort of overall view of photosynthesis. And in fact, the Calvin cycle requires more ATP than NADPH. And ATP is also used in other components of the chloroplasts, other reactions as well. So, so there has to be the ability to synthesize a large amount of ATP. And so the only way that chloroplasts can do that is if they build up these hydrogen ions in the lumen. And so there's a couple of additional ways aside from transporting across by PQH2. So we're going to take a look at that next, and that's called the PQ cycle. So we're going to take a minute to kind of describe this Q cycle. And the goal of the Q cycle is to transport more hydrogen ions across the thylakoid membrane from the stroma into the lumen. So right at that point where we're going to do our best here to draw all this out. So let's say that we're drawing here the cytochrome B6F complex. We've got this binding site here called cytochrome B6. So here's the cytochrome complex. Alright, so within the thylakoid membrane we've got our first PQH2 that's diffusing through the membrane, and it reaches the cytochrome F site where the iron nearby it reaches the iron sulfur complex in the cytochrome complex here. And it's going to transfer one of its electrons to that iron sulfur complex, reducing it, as we said earlier. And then that one electron is then going to be transferred to plastocyanin, where the plastocyanin becomes reduced, and off it goes to photosystem 1. All right, the second electron plus, okay, first of all, when this plastocyanin, or the plastoquinone, sorry, gets oxidized, it's now going to become a PQ, and is going to then sit up here in this cytochrome B6 site. So then the second electron is going to join it, that second electron that came from the first PQH2, which is going to make this become a PQ minus. And it's going to sit in that site and kind of hang out until a second PQH2 comes along. Now when this plastoquinone becomes oxidized, remember it's going to release 2 hydrogen ions into the lumen. And that's the whole goal here, is to accumulate hydrogen ions in the lumen. Now I've tried to carefully erase what we needed so that we could show what happens here when the second plastoquinone comes along. So here's the second plastoquinone that's diffusing through the membrane, reaching the cytochrome complex. It has one electron again that it transfers to the iron sulfur complex, which then is used to reduce plastocyanin again, and off it goes to photosystem 1. And then remember, it's got a second electron now that it's going to add to this waiting plastoquinone in the cytochrome B6 site. And so that's now going to become PQH2 once again, because it's going to pick up 2 more. Once it has that second electron, it's going to pick up 2 more hydrogen ions from the stroma, add to the PQ, basically minus 2, to become PQH2. Then that PQH2 dissociates from the cytochrome B6 site and is now back in the thylakoid membrane, or in the what we call the PQH2 PQ pool in the membrane. So plastoquinone can be reduced essentially in the Q cycle. This is all the Q cycle. So one thing to remember is that when that second PQH2 comes along and is oxidized, it too is releasing 2 hydrogen ions into the lumen, as we saw with the first PQH2 released 2 hydrogen ions into the lumen, for a total of 4 hydrogen ions in the lumen between the first PQH2 and the second one. But now remember, we've got that first PQH2 that was sitting here waiting. It picked up 2 more hydrogen ions and re-enters the pool, and that actually is going to come back around through the same procedure here and ultimately give 2 more hydrogen ions. So it can actually appear like there's a total of 6 hydrogen ions, but remember, these 2 additional ones would not associated with the first PQH2 would not have been possible without the second PQH2 donating its one of its electrons back to the first PQ. So we have a net transfer of 4 hydrogen ions per PQH2 molecule that gets oxidized because of the Q cycle. Okay, so it may be worth kind of stopping and going back through the series of steps that we looked at in these with the oxidation of 2 separate PQH2 or plastoquinol molecules to result in 4 hydrogen ions moved into the lumen per PQH2 oxidized. So this, again, ultimately, Q cycle is a way to add more hydrogen ions to the lumen, which ultimately is going to benefit the plant, or the yield benefits towards producing more ATP. So another method here that we want to emphasize that helps to accumulate hydrogen ions in the lumen is demonstrated in this second figure over here. So over here on the left, we have what's considered our standard Z scheme, showing the free energy that results from these various redox reactions. And so you can see water is a relatively low energy molecule, and through a series of redox through the photooxidation step where electrons are added to P680, and then that P680 electron gets excited, it reaches a higher energy level, and you can see all the steps here with a little bit of loss of energy along each step, and a second photooxidation step as a result of the excited electron in P700, ultimately to transfer those electrons to NADPH. So we call, this is the Z scheme that illustrates linear electron flow, whereas over here, in a linear electron flow, we have the accumulation of protons into the lumen, as you can see illustrated here, as well as the Q cycle that we just talked about that would add add hydrogen ions to the lumen. Now over here we have what's called cyclic electron flow, and which means that we can also call this non-cyclic electron flow, where we have where we see that in PS1, photosystem 1, when the electron is used to oxidize ferredoxin right here, instead of reducing NADP+, this ferredoxin is going to circle back around in the stroma and reduce another plastoquinone. And by reducing another plastoquinone, that plastoquinone enters the plastoquinone pool again in the thylakoid membrane and can be sent on to the cytochrome B6F complex and become oxidized, and in that process adding more hydrogen ions to the lumen. So we'll expand what we see in this diagram down in the next diagram here. And in this diagram, we're looking at the electron transport chain, but this is cyclic electron flow, where we can see the electron that is excited in photosystem 1, here's photosystem 1, is used to reduce ferredoxin. It receives 2 electrons, remember, and those 2 electrons then, instead of being used to reduce NADP+ to NADPH, the ferredoxin as a mobile electron acceptor carries those electrons over to the plastoquinone that's waiting here at the cytochrome B6F complex. Once the plastoquinone is reduced, it picks up 2 more hydrogen ions. Remember, that's kind of the idea here. Re-enters the thylakoid plastoquinone pool, and then can come around to the iron-sulfur complex and be re-oxidized. When the plastoquinone gets re-oxidized, releasing its electrons again to iron-sulfur compound and plastocyanin, it releases those 2 hydrogen ions into the lumen. So essentially it's transported 2 hydrogen ions from the stroma into the lumen, thereby increasing the concentration of hydrogen ions. And of course that's going to only benefit ATP production because it relies on that concentration gradient, which we can see next.